Hello, my name is Kyle Boudreau, and I'm going to be presenting my TEDdy talk on chytridiomycosis effect on skin microbiota. Now, it's worth noting that there have been extensive studies that have looked at human microbiota and human health. But what about other animals? What studies exist out there that have especially maybe looked at serious health impacts regarding microbiome, especially something like dysbiosis and animal health? And this led me to think about particularly chytrid. Chytrid is a infection that has been studied quite extensively and has had serious global impact. So what could potentially be a link between chytrid infection or chytridiomycosis and the skin microbiome? Some quick background about chytridiomycosis. So this has spread globally, at least over the past few decades, and has led to an estimated 700 amphibian species, a decline in an estimated 700 amphibian species, and by some estimates, maybe tens or more extinctions as well. So chytridiomycosis is caused by chytrids, which are a member of the Chytridiomycota group. And particularly one species has gotten the most study because this is the one that especially has caused the greatest impact, which is Batrachochytridium dendrobatitis, or BD as I'm going to refer to it as. So where has this had impact? We can see here in the Western Hemisphere, particularly North America, along the west side or the west coast, California especially has been severely impacted, as well as places like Oregon and Washington and Idaho. In the middle here of the Western Hemisphere, Yucatan Peninsula, Central America, also parts of South America have also tested positive sites with BD presence. The other side of the globe, Japan especially, has been severely impacted by BD presence as well. Here's a photo of mountain yellow leg frogs, or Rana mucosa, which have been killed by chytrid. So this is especially one species that has been susceptible and severely impacted by the BD presence. So the life cycle, this is a flagellated zoospore. At least part of its life cycle exists as this aquatic motile zoospore that has a flagellum. And this is especially where amphibian species are going to be most susceptible. Amphibian species obviously spend part of their life cycle in aquatic environments, whether it be as adults breeding or as larvae. So of course, they are particularly sensitive. And this chytrid, this zoospore, infects skin cells, so it particularly likes keratinized epithelial cells. So this study that was done by Sun et al. Looked at the associations of BD with skin bacteria and fungi on Asian amphibian hosts. They examined 4 anurans, 3 frog species and 1 toad species, in the Guangxi province of southern China. Asia has not gotten a lot of study regarding BD infection in amphibian hosts, so this is where they wanted to focus their study. What was noteworthy is that there was particularly a correlation between something like bacterial richness and infection intensity. So we can see at least in 2 species here, there is a correlation between the infection intensity, which they determined as the number of reads of BD in individuals, versus the bacterial richness. And at least in one example, one species, we can see that there was also a link between the diversity of bacteria and the intensity as well. What they did also find is that of the 4 that they sampled, 68 of these amplicon sequence variants that were determined were shared among all 4 of these frog species. And this led them to think that there must be something about these 68 species that might be connected to providing some type of mechanism that affords protection against BD infection. The other important thing that they found is that there's a link between what they are determining anti-BD bacterial richness and prevalence, as we can see here in figure B, as well as infection intensity and antibacterial BD richness as well. So they concluded BD infection prevalence, intensity, and anti-BD relative abundance are correlated, and particularly, again, they're looking at these 68 species that they determined are common among all 4 groups. The other important thing that's worth noting is that they also found, at least in 2 species, that Prevotella, there is a positive correlation to Prevotella and BD infection. And there's also a negative correlation between Ascomycota fungal species, at least in 2 species again, and BD infection as well. So this is based off of presence of BD, and they saw an increase or positivity with Prevotella and a negativity or decrease in Ascomycota. So the key takeaways from this study Evidence that infection intensity and bacterial richness are correlated. So the intensity of the BD infection is correlated to bacterial richness. Infection intensity and anti-BD relative abundance. So there's a positive correlation between the anti-BD relative abundance and the infection intensity. And there also is some evidence that there are species, particularly bacterial species, that appear to help modulate the infection. So what is it about that, particularly, that is causing that? And lastly, Prevotella. I chose Prevotella because Prevotella is something that gets talked about a lot, especially regarding human health, because often Prevotella is shown to be correlated with especially inflammatory diseases. So this is something that definitely shows up a lot. And here we can see it showing up in amphibian species regarding skin microbiota and its positive correlation with the increase of BD infection. And this led them to believe that there is possibly maybe an increased sensitivity with the presence of Prevotella to BD infection in these frog species. So what's next? Where do we go from here? So larger studies need to examine skin microbiota between infected and non-infected amphibians. The limits of this particular study is that they unfortunately did not collect enough of these non-infected amphibians, individuals of these 4 species, with the exception of one, which was the T. rhabdodiscus. And even then, it was only about 20 individuals that they're able to sample. So we need a larger study that looks at infected versus non-infected, especially in amphibians. How are so-called anti-BD species preventing infection, if they are indeed helping to inhibit BD growth or BD infection of the skin cells? What is it about it? Are they producing some type of antimicrobial agent, something that could possibly be used in therapeutics or as preventative treatment? And lastly, where do we go from here? Where can we expand this research? What can we do with this knowledge? How can we, especially regarding chytrid impact and its effect globally, how can we use this data to help further try to thwart the impact that chytrid infection or chytridiomycosis is having on the amphibian species across the globe? All right, thank you very much. Hopefully you found this very interesting. I know I did.